Good afternoon. Today I would like to introduce you the next pharmacology lecture, drugs acting on autonomic nervous system, specifically cholinergic drugs and anticholinergic drugs. Lecture plan: general considerations, direct acting cholinergic drugs, indirect acting cholinergic drugs, anticholinergics. Let's start from general considerations. Classification of nervous system. As you remember, the nervous system is a complex network of nerves and cells that carry messages to and from the brain and spinal cord to various parts of the body. The nervous system includes both the central nervous system and peripheral nervous system. The central nervous system is made up of the brain and spinal cord. The peripheral nervous system is made up of the somatic and autonomic nervous system. Today we will talk about peripheral nervous system, specifically parasympathetic nervous system, which is the part of autonomic nervous system. Neurons in the peripheral nervous system. The neuron is the basic unit in the nervous system. It is a specialized conductor cell that receives and transmits electrochemical nerve impulses. A typical neuron has a cell body and long arms that conduct impulses from one body part to another body part. For each of the chain of impulses, there is one preganglionic neuron, or one before the cell body or ganglion, that is like a central controlling body for numerous neurons going out peripherally. The preganglionic neuron is located in either the brain or the spinal cord. In the autonomic nervous system, this preganglionic neuron projects to an autonomic ganglion. The postganglionic neuron then projects to the target organ. In the somatic nervous system, there is only one neuron between the central nervous system and the target organ, while the autonomic nervous system uses two neurons. Somatic nervous system. The somatic nervous system consists of peripheral nerve fibers that pick up sensory information or sensations from the peripheral or distant organs and carry them to the central nervous system. These also consist of motor nerve fibers that come out of the brain and take the messages for movement and necessary action to the skeletal muscles. For example, on touching a hot object, the sensory nerves carry information about the heat to the brain, which in turn, via the motor nerves, tells the muscles of the hand to withdraw it immediately. The whole process takes less than a second to happen. The cell body of the neuron that carries the information often lies within the brain or spinal cord and projects directly to a skeletal muscle. Autonomic nervous system. Another part of the nervous system is autonomic nervous system. It has three parts: the sympathetic nervous system, the parasympathetic nervous system, and the enteric nervous system. This nervous system controls the nerves of the inner organs of the body on which humans have no conscious control. This includes the heartbeat, digestion, breathing, etc. The nerves of the autonomic nervous system innervate the smooth involuntary muscles of the internal organs and glands and cause them to function and secrete their enzymes, etc. The enteric nervous system is the third part of the autonomic nervous system. The enteric nervous system is the complex network of nerve fibers that innervate the organs within the abdomen, like the gastrointestinal tract, pancreas, gallbladder, etc. It contains nearly 100 million nerves. On this picture you can see parasympathetic nerves and sympathetic nerves and some of the functions on different organs which they provide. And in here you can see the general outlay of afferent autonomic nervous system. The transmitter released and the primary postjunctional receptor subtype is shown at each synapse near a effector junction. For example, acetylcholine, noradrenaline, nicotinic, muscarinic, alpha and beta receptors. About them we will talk a little later. And on this slide, some differences between somatic and autonomic nervous system. For example, number of neurons in the afferent pathway. In somatic it is 1, in autonomic it is 2. Neurotransmitter receptor at neuron target synapse. For somatic it is acetylcholine, nicotinic, and autonomic it is acetylcholine, muscarinic, or norepinephrine, alpha or beta. Target tissues. In somatic, it is skeletal muscles. In autonomic, it is smooth and cardiac muscle, some endocrine and exocrine glands, some adipose tissue, and others. As I mentioned before, autonomic nervous system is divided into sympathetic and parasympathetic. What are the differences in sympathetic and parasympathetic divisions? They come from the different regions of the CNS, sympathetic from the thoracolumbar region, parasympathetic from the craniosacral region, different locations of ganglia, sympathetic close to spinal cord in a chain, parasympathetic close to target organs, differing length of postganglionic fibers, sympathetic long, parasympathetic short, postganglionic branching, in sympathetic lots, so the multiple organs can be mobilized at once, in parasympathetic, very little branching. On this picture, I would like you to remember the effects of sympathetic and parasympathetic stimulations. As a rule, they are antagonistic. For example, heart, effect of sympathetic stimulation is increased heart rate, increased force of contraction. Effect of parasympathetic stimulation, decreased heart rate, decreased force of contraction. For example, digestive tract, effect of sympathetic stimulation is decreased motility, contraction of sphincters, inhibition of digestive secretions. Effect of parasympathetic stimulation, increased motility, relaxation of sphincters, stimulation of digestive secretions. Eye, dilation of pupil, adjustment of eye for far vision in cases of sympathetic stimulation, constriction of pupil, adjustment of eye for near vision in cases of parasympathetic stimulation. Excitatory and inhibitory neurohumoral transmission. Whether the result of synaptic transmission will be excitatory or inhibitory depends on the type of neurotransmitter used and the ion channel receptors they interact with. Different neurotransmitters can be very similar in structure, but can have profoundly different functions. And in here you can see diagrammatic representation of steps in excitation and inhibitory neurohumoral transmission. Termination of transmitter action. Parasympathetic neuron. The primary transmitter acetylcholine is rapidly hydrolyzed by a specific enzyme, acetylcholinesterase, located strategically on the synaptic membrane. A common co-transmitter is vasoactive intestinal peptide, which on release diffuses slowly to be degraded by peptidases at distant sites. It may act on the same as well as neighboring effectors. Sympathetic neuron, the primary transmitter noradrenaline, is largely taken back into the neuron by membrane-bound norepinephrine transporter, NET, and recycled. A minor fraction diffuses away. One of the co-transmitters is neuropeptide Y, NPY, which on release meets the same fate as VIP. GABAergic neuron, the amino acid transmitter gamma-aminobutyric acid, released into the synaptic cleft, is partly taken up into the neuron by GABA transporter as well as into neighboring glial cells. Some of it dissipates by diffusion. Co-transmission. It has now become clear that the classical one neuron, one transmitter model is an oversimplification. Co-transmission. The co-transmitter is stored in the prejunctional nerve terminal along with the primary transmitter, but in separate vesicles, in some cases in the same vesicle itself. Nerve impulse releases both the transmitters concurrently. Acting on its own receptors, the co-transmitter modifies responsiveness of the effector to the primary transmitter or substitutes for it. Co-transmitter may also act on prejunctional receptors and modulate release of the transmitters. In autonomic nervous system, besides the primary transmitters acetylcholine and noradrenaline, have been elaborate purines, peptides, nitric oxide, and prostaglandins. Receptors main groups. First of all, to remind you, what does it mean receptor? Receptors are chemical structures composed of protein that receive and transduce signals that may be integrated into biological systems. These signals are typically chemical messengers which bind to a receptor. They cause some form of cellular tissue response, for example, a change in the electrical activity of a cell. A receptor is a protein molecule that recognizes and responds to endogenous chemical signals. For example, an acetylcholine receptor recognizes and responds to its endogenous ligand, acetylcholine. Examples of receptors: cholinergic receptors from M1 to M5, also N1 or NN, N2 or NM, adrenergic receptors alpha-1, alpha-2, beta-1, beta-2, beta-3, dopamine receptors from D1 to D5, serotonin receptors, GABA receptors, benzodiazepine receptors, histamine receptors, cannabinoid receptors, opioid receptors, and others. Subunits of cholinergic channel receptor. Cholinergic receptor consists of 5 protein subunits which are located surround the sodium channel. And on this picture you can see cholinergic channel function when it is inactive and activated. Next one is synaptic cleft. Synaptic cleft, the space between neurons at a nerve synapse across which a nerve impulse is transmitted by a neurotransmitter. Neurotransmitters are released into a synapse in packaged vesicles called quanta. One quantum generates what is known as a miniature end plate potential, MEPP, which is the smallest amount of stimulation that one neuron can send to another neuron. And in here you can see the literature link of an article of M. Simonu, L. Tauch, and GBU about quantal release of acetylcholine and its analysis. Neurotransmitter acetylcholine. Acetylcholine, a chemical message released by nerve cells to send signals to other cells. Its name is derived from its chemical structure. It is an ester of acetic acid and choline. Parts in the body that use or are affected by acetylcholine are referred to as cholinergic. Substances that interfere with acetylcholine activity are called anticholinergics or cholinoblockers, cholinergic antagonists. Source tissues: motor neurons, parasympathetic nervous system, brain. Target tissues: skeletal muscles, brain, other organs. Receptors: nicotinic, muscarinic. Agonists: nicotine, muscarine, cholinesterase inhibitors. Antagonists: tuberculin, atropine. Precursor: choline acetyl coenzyme A. Biosynthesis: choline acetyltransferase. Metabolism: acetylcholine esterase. On this slide you can see synthesis, storage, and destruction of acetylcholine. What are the steps? First of all, synthesis of acetylcholine itself. Second, uptake to storage vesicles. Third, release of this neurotransmitter. Fourth, its binding to the receptor. Fifth, degradation of acetylcholine with the help of acetylcholine esterase. And sixth, recycling of choline. Choline is going back. Classification of cholinergic receptors. Cholinergic receptors are classified into 2 big groups: nicotinic receptors and muscarinic receptors. Nicotinic receptors are subdivided into N1 or NM and N2 or NN. Muscarinic receptors are subdivided into M1, M2, M3, M4, M5. In more details, N1 or NM, neuromuscular junction; N2 or NN, autonomic ganglia, central nervous system, adrenal medulla; M1, striatum, cortex, hippocampus; M2, forebrain, thalamus, heart, pupil, spinal cord, exocrine; M3, brain, hypothalamus, pupils, exocrine, peripheral arteries; M4, striatum, cortex, hippocampus, spinal cord; and M5, dopaminergic neurons, basal ganglia, brain vasculature. The next part of our lecture is direct-acting cholinergic drugs. As you remember, one of the types of cholinergic receptors are called as muscarinic. They are called in the name of muscarine, which is found in mushrooms, Amanita muscaria. And here you can see any information about these types of mushrooms, but I would like to point your attention that muscarine is non-selective agonist of the muscarinic acetylcholine receptors, and it is not used clinically. Muscarine, it is a component which is causing poisoning in cases of ingestion of the mentioned mushrooms. On this slide, you can see subtypes of muscarinic receptors which are well known: M1, M2, M3. Their location and function, nature, transducer mechanism, agonists and antagonists. Next one is nicotine. Nicotinic receptors are called in the name of nicotine. Nicotine is found in tobacco. It acts as a receptor agonist at most nicotinic acetylcholine receptors, except at two nicotinic receptor subunits, where it acts as a receptor antagonist. And on this slide, you can see subtypes of nicotinic receptors, typical locations, responses, mechanism, prototype agonist, and prototype antagonist. The first group of cholinergic drugs is cholinomimetics or parasympathomimetics. These are the drugs which are acting non-selectively on cholinergic receptors. These are the examples: acetylcholine, metacholine, carbachol, beta-nicol. The next one is M-cholinomimetics. These drugs are acting selectively on M-cholinergic receptors, pilocarpine and aceclidine. And also below you can see the drug forms of these drugs. The next one is N-cholinomimetics. These drugs are acting selectively on N-cholinergic receptors. For example, lobeline and cititone, lobesyl tabex, nicotine, varenicline, bupropion. And below you can see the examples of their doses and drug forms. General pharmacological actions of cholinergic drugs. Muscarinic, affect heart, bradycardia, blood vessels, endothelia-dependent relaxing factor, smooth muscles, contraction of most smooth muscles, glands, secretion increasing, eye, meiosis, circular muscle contraction. Nicotinic, autonomic ganglia, stimulated, skeletal muscles, contraction of muscle fibers, respiratory system, respiratory analeptics. The next part of our lecture is indirect acting cholinergic drugs. Indirect acting cholinergic drugs are called as anticholinesterases. These drugs are inhibiting acetylcholinesterase, and as the result, the activity of natural acetylcholine is increased. They are categorized into reversible anticholinesterases and irreversible anticholinesterases. Reversible anticholinesterases are subdivided into carbamates, physostigmine, neostigmine, pyridostigmine, ribostigmine, edrophonium, galantamine, and acridine, tacrine. Mechanism of action of these drugs. Enzyme inhibition. They inhibit the enzyme acetylcholinesterase reversibly, results in greater availability of acetylcholine at its sites of action. Presynaptic effects. In the absence of non-depolarizing neuromuscular blocking drugs, administration of anticholinesterase drug may produce spontaneous contractions of skeletal muscles. These presynaptic effects are abolished by a small dose of non-depolarizing neuromuscular blocking drug, suggesting that acetylcholine receptors are involved, and direct effects on the neuromuscular junction. On this slide, you can see the comparative features of two anticholinesterase drugs, which are physostigmine and neostigmine. The source, chemistry, oral absorption, CNS actions, and others. The second group of anticholinesterase drugs is irreversible anticholinesterases. They are subdivided into organophosphates, diflos, ecotiophate, diacylone, tabun, serine, soman, and carbamates, carbaryl, propuxur. Please note that these drugs are insecticides and nerve gases for chemical warfare. They are inhibiting the activity of acetylcholine esterase irreversibly, and most of them are not used clinically. Pharmacological actions of anticholinesterases. Ganglia, blockade of transmission due to depolarization. CVS, bradycardia and hypotension. Skeletal muscles, persistent depolarization. CNS, altering response if penetrate into brain. Other effects, stimulation as for acetylcholine. Uses of anticholinesterases. As miotic for glaucoma, myasthenia gravis, postoperative paralytic ileus urinary, postoperative decurarization, cobra bite, belladonna poisoning, other drug overdosage, Alzheimer's disease. And on this slide you can see differences between the two types of cholinesterases. Acetylcholinesterase, which is true, and butyrylcholinesterase, which is pseudocholinesterase. Distribution, hydrolysis, inhibition, and function. And the last part of our lecture is anticholinergic drugs. Classification of anticholinergics. We may also call them cholinergic antagonists or cholinoblockers, depending on the type of receptors which they are blocking. They are divided into muscarinic antagonists, nicotinic antagonists. Muscarinic antagonists are subdivided into selective and non-selective. Nicotinic antagonists are subdivided into ganglionic blockers and neuromuscular blockers. Mechanism of action. Anticholinergic drugs are those which antagonize the effect of neurotransmitter acetylcholine on autonomic effectors and in the CNS exerted through muscarinic receptors. Though nicotinic antagonists also block certain actions of acetylcholine, they are referred to as ganglion blockers and neuromuscular blockers. Muscarinic receptor sites, heart, salivary glands, smooth muscles of GIT, genitourinary tract, urinary bladder. The first group of anticholinergic drugs is M-cholinoblockers or muscarinic receptor antagonists. They are divided to, first, natural alkaloids, atropine, scopolamine, second, semi-synthetic derivatives, homatropine, atropine metonitrate, hyoscine butyl bromide, tiotropium bromide, and third, synthetic compounds, which are subdivided into mydriatics, antisecretory antispasmodics, vasoconselectives, and antiparkinsonian. Atropine as the prototype of M-cholinoblockers. Atropine is the medication used to treat certain types of nerve agent and pesticide poisonings, as well as some types of slow heart rate and to decrease saliva production during surgery. It is typically given intravenously or by injection into a muscle. Eye drops are also available, which are used to treat uveitis and early amblyopia. The intravenous solution usually begins working within a minute and lasts half an hour to an hour. Large doses may be required to treat some poisonings. Common side effects include dry mouth, large pupils, urinary retention, constipation, and a fast heart rate. It should generally not be used in people with angle closure glaucoma. While there is no evidence that its use during pregnancy causes birth defects, it has not been well studied. It is an antimuscarinic, a type of anticholinergic, that works by inhibiting the parasympathetic nervous system. Atropine occurs naturally in a number of plants of the nightshade family, including deadly nightshade, Jimson weed, and mandrake. It was first isolated in 1833. It is in the World Health Organization's list of essential medicines, the most effective and safe medicines needed in a health system. And below you can see the picture of Atropa belladonna, which includes this atropine. On this slide you can see atropine substitutes, quaternary compounds. Hyoscine butyl bromide, atropine metonitrate, ipratropium bromide, tiotropium bromide, and others. In here you can see tertiary amines, dicyclomine, valethamate, and pyrenzepine. and other substitutes, for example, vasoconstrictive oxybutynin, mydriatics, homatropine, cyclopentolate, and tropicamide, and antiparkinsonian, trihexyphenidyl, procyclidine, orphanadrine, and promethazine. Target of pharmacological action of anticholinergic drugs: CNS, CVS, eyes, smooth muscles, glands, body temperature. Uses of M-choline blockers: as antisecretory, as antispasmodic, bronchial asthma COPD, as mydriatic and cycloplegic, as cardiac vagolytic, for central action, Parkinsonism, motion sickness, antagonize muscarinic effects of drugs and poisons. The next pharmacological group is skeletal muscle relaxants. They are divided into neuromuscular blocking agents and directly acting agents. Neuromuscular blocking agents are subdivided into non-depolarizing and depolarizing ones. Non-depolarizing are competitive blockers, pachycurare. They are acetylcholine concurrent. For example, tubocurarine, diplacine, tercuronium, pancuronium, vecuronium, and others. The antagonist of them is prozerine, or in other words, neostigmine. It is cholinesterase blocker. Depolarizing, leptocurare, suxametonium, and decametonium. They are metabolized by pseudocholine esterase. And second group, directly acting agents, are dantrolene sodium and quinine. The explanation about them will be in the next slides. Non-depolarizing or competitive blockers can be also classified into long-acting, intermediate-acting, and short-acting. Long-acting examples you can see in here, which are tubocurarine chloride, pancuronium, doxacuronium, and pipecuronium. Intermediate acting are vecuronium, atracurium, cisatracurium, rocuronium, and rapacuronium. Short acting is mivacurium. Depolarizing blockers, as I mentioned before, succinylcholine or suxametonium and decametonium. Below you can see the formulas of these agents. On this slide, you can see the differences in actions of competitive and depolarizing neuromuscular blocking agents. For example, tubocurarine, which is non-depolarizing competitive blocker, it produces progressive decrease in twitch tension. Tetanic stimulation produces poorly sustained contracture, which is followed by post-tetanic potentiation. Neostigmine, anticholinesterase drug, restores the twitch contractions. Succinylcholine, which is depolarizing blocker, produces initial augmentation of twitches followed by progressive block. Tetanus is well sustained, but there is no post-tetanic potentiation. Block is not reversed by neostigmine. It is even rather worsened. A tetanic contraction is a sustained muscle contraction evoked when the motor nerve that innervates a skeletal muscle emits action potentials at a very high rate. During this state, a motor unit has been maximally stimulated by its motor neuron and remains that way for some time. This occurs when a muscle's motor unit is stimulated by multiple impulses at a sufficiently high frequency. Each stimulus causes a twitch. The second group of skeletal muscle relaxants is directly acting skeletal muscle relaxants. These are dantrolene sodium and quinine. Dantrolene sodium acts on the ryanodine receptor calcium channels in the sarcoplasmic reticulum of the skeletal muscles and prevents calcium-induced calcium release through these channels. Intracellular release of calcium needed for excitation contraction. Quinine increase refractory period and decrease excitability of motor end plates. Thus, responses to repetitive nerve stimulation are reduced. It decreases muscle tone in myotonia congenita. And on this slide you can see comparative properties of neuromuscular blocking drugs. For example, long-acting tubocurarine, pancuronium, doxacurium, pipecuronium, intermediate-acting vecuronium, atracurium, cisatracurium, and rocuronium, short-acting mivacurium and succinylcholine. Their doses, onsets, durations, histamine release, ganglionic block, and vagal block. The next pharmacological group is ganglioblockers. We may also call them ganglionic blockers or NN receptor blockers. They are divided to competitive blockers and persistent depolarizing blockers. Competitive blockers are subdivided into quaternary ammonium compounds, for example hexametonium and pentolinium, and amines, for example mecamylamine, pempidine. On this slide you can see examples of ganglionic blockers, their drug forms and doses. This table explains us relative autonomic tone and effects of ganglionic blockage on organ function. For example, organ heart, dominant tone parasympathetic, effect of ganglionic blockage will be tachycardia. Blood vessels, dominant tone sympathetic, effect of ganglionic blockage will be vasodilation, abolition of reflexes. Iris, dominant tone parasympathetic, effect of ganglionic blockage will be mydriasis. Ciliary muscle, dominant tone parasympathetic, effect of ganglionic blockage will be cycloplegia, and others. And as the additional part of our lecture, I would like to discuss with you some examples of MCQs. An epileptic remedy of reflective type from the anticholinogenics group was given to the patient for restoration of breathing after poisoning with carbon monoxide. What medicine was prescribed to the patient? According to the drugs which we have in the options, the only one which is anticholinergic is lobeline hydrochloride. A patient with drug intoxication presented with the dryness of oral mucous membrane and mydriatic pupils. Such action of this drug is associated with the following effect. The mentioned manifestations, which are dryness of oral mucous membrane and mydriatic pupils, are pointing that the correct answer will be muscarinic cholinereceptor block. A patient complains of dizziness, thirst, difficult swallowing, and impaired vision of close objects. Objectively, respiratory rate is increased, pupils are dilated, general agitation, talkativeness, though the speech is indistinct. Blood pressure is 110 systolic, 70 diastolic. Heart rate is 110 beats per minute. Given symptoms can indicate overdosage of the following drug. Please memorize these manifestations because these are the symptoms of overdosage of atropine. After anephrectomy, the patient developed enteroparesis. What cholinergic agent with anticholinesterase action should be prescribed in this case? In the options, the only one anticholinesterase agent is present, which is prozerine, and please remember that prozerine has got the other name, which is neostigmine. Thank you for the attention.